Okay, Carl Pilcher, director, and Ed Gulish, deputy, are both on travel and have asked me to introduce the seminar today. We're fortunate in having six summer interns from the Astrobiology Center at Goddard here today to give brief summaries of the research that they performed this summer. This is our fourth summer running this internship program, which we're happy to say has been wildly successful, this summer being no exception, and I think it's safe to say that each and every intern that's gone through here has actually accomplished some new state-of-the-art research. And of course, the most important thing for each person is that they've experienced the manner in which state-of-the-art research is done, and that's why they're here. So without further ado, let me call on our first intern, Charlotte Karlstrom, who is from Emory University, who's been working this summer with Jen Eigenbrode, a member of our astrobiology team, and she will speak to us today. Charlotte will speak to us on molecular signatures of life in surface ice and snow as a Mars analog. Charlotte?
Okay, so my name is Charlotte Karlstrom, and So my summer project dealt with trying to find signatures of life in surface ice and snow so that we can take whatever we learn about signatures of life here on Earth to other places where life isn't apparent, like Mars or any other icy moon, or, well, it doesn't have to be icy, but any other planet really. So not only do we have to find what these signatures of life are, but we have to be able to determine how to examine them in another place. And so after we find the signatures, we have to compare the effectiveness of pyrolysis, which is what SAM, the Sample Analysis and Mars instrument, and Phoenix, which is actually there right now, the effectiveness of those methods versus what we use conventionally, which is the lipid extraction and derivatization. And my work here was directly related to SAM in particular because of the GCMS that we use is the same setup as SAM, as is the pyrolysis cell is set up just like SAM with the trap, with the glass beads and tinacs. It's the exact same setup, so this is why it's related to that. So what are lipids? Lipids are fatty acids and their derivatives. For the purpose of this, is going to be just fats. But they're found within cell membranes, as you can see here, and they're also used for cellular structure, cellular messengers, and energy storage. So the picture that you see here are three of the most widely distributed fatty acids, and these are important because they're found in pretty much all life, but they're signatures of life in themselves, in that they're even chained, which is a signature of life because of lipogenesis, how lipids are made. It starts with acetic acid, which is a 2-carbon chain. So usually you get even numbers of carbons. But also linoleic acid in the bottom shows unsaturation, which is also a trademark of life, because usually when you see unsaturated fatty acids, they have to be confined to some form of life so that they stay in the unsaturated state. This is a site in Norway where the samples were collected. And here in the top left, you see the green snow algae. Here in the background where I'm pointing is the red snow algae. And here is the cryoconite, which usually form in the summer. And they form when matter gets collected, dark matter gets collected in the ice and then absorbs heat from the sun, and it starts sinking in. And eventually they become harvesters of life because they have lots of nutrients and good stuff where things like to grow. This is a close-up microscopic images of the snow algae samples. These were taken, I think, 2 years before the samples that I'm analyzing. But these are red snow algae samples. These two are red snow algae. Here you see the orange, the yellow, the red, the green, and these are mineral grains. Here you see the green and the red. Here's red and orange. And here, these are Calamidomonas nivalis, which is the most abundant snow algae. And this is a green one, and this is a red, and this is actually pointing to the chlorophyll in both of them, so you can see the difference. This is the setup of the instrument and the methodology that we use. So there's two different methods. The first one is the conventional, and that's the wet chemistry method, and that uses the accelerated solvent extractor uses high temperatures and pressures to extract lipids from the cells. And these are then methylated with acetylchlorine and methanol at 100 degrees, and this goes directly into the GC through here, where they're separated and then analyzed in the mass spec. The second method, which is what Sam and Phoenix use, is pyrolysis, and that takes a sample and heats it to thermally evolve whatever is found in there, and then that goes through the GC column and is analyzed. So those are the two methods that we're comparing right now. This is what I started with to try to learn how to read mass spectra as well as gas chromatograms. And this is the fatty acid methyl ester standard. Each of the peaks represents a different compound. The one that I have highlighted here at 44 minutes turns out to be hexadecanoic acid, which is the most widely distributed fatty acid again. And so this around here is basically another dimension to this, the gas chromatogram. And it shows that the presence of this, and you can tell by its weight, which is 270, that's the molecular ion right here. But each of the peaks is representative of a fragment of this molecule, depending on how it breaks up. So when you have only the part on the right with one carbon, you get the 74. When you have two carbons, you get the 87, and so on and so forth. And these peaks, the gas chromatograph peaks are important because you can either refute or support the identity of an unknown based on the elution time, because it's the same every time, based on that it's on the same method. This is the actual data of one of the samples. This is the green algae. And on the top you see the conventional method, and it's very clean, and you can see each of the acids that we were expecting to find. And these are actually found in all samples, not just in this one, the ones you see here. When you go to pyrolysis, which actually was at 610 degrees Celsius, you don't see a clear definition, I guess, of the peaks that we were looking for. And here I found a lot of sugars and a lot of different things that not necessarily were expected to be found in the algae. Lots of sugars and unusual rings present. And wait, let me go back. We think that this could be also because the trap that the instrument has, the pyrolysis cell, could have retained the fatty acid. So another option for Sam is to get rid of the glass bead trap and the Tenax to try to get all of the gas that we get. So that's something that could be done to try to see if we see the same fatty acids that we know are present. So these are all the fatty acids that were found in the snow algae samples using the conventional method. And as you can see, there's plenty of unsaturation here, which we expected to find. Most of them are even chained, which is also something we expected. And something interesting that we found is this compound here at dodecane 1,1 is actually a diol, not methoxy, because this is, once it's been derivatized, it changes to the OCH3 instead of the OH. And that hasn't been found before, and it's interesting because we don't really know what it's doing. So that's something that needs to be looked at too. In conclusion, we determined that wet chemistry is a lot cleaner and clearer than its pyrolysis. But since pyrolysis is what most of the instruments nowadays are using, it has to be taken into account. It has to work. It has to be worked with a little more to try to find how it matches the wet chemistry data. We also found that there are several fatty acids that serve as biomarkers for life identification, which is a good thing. And we found that those tend to agree with the even chain fatty acid theory of life, how lipogenesis is made, how lipids are made, the 2-carbon chain. And also the unsaturation, we found quite a few unsaturated fatty acids. And so I would like to thank GCA for a great summer, Dr. Jen Eigenbrod for a lot of learning, Dr. Ingeton Kate and Dr. Paul Mahaffey for all their help while Jen's been gone to Norway this year, Dr. Michael Muma for GCA support, and Ms. Corinne Evie for everything she's done for us this summer. And that's it.
Take a question or two?
Sure.
Good. Okay, let's go first to anyone in the field and on remote sites and other people out there who'd like to make a comment or ask a question. Oh, Charlotte? A passive group. Okay, how about here in the room? I would like to know why you found it surprising that you found sugars inside some of your algae.
Well, it's not surprising that they're found in what we analyze. It's surprising that they're inside, considering that, well, we were looking for lipids in particular, and those tend to give a strong signal, and we didn't see that, but we saw the sugars, and those could be not necessarily in the algae, but anywhere within the sample, since those weren't particularly extracted and analyzed just for that.
So there could have been contamination then.
Not contamination necessarily, but sources that are non-algae, if you call that contamination, I guess. I mean, it's their environment, I guess.
All right, any other questions? Okay, let's go on. Our next speaker will be Ariel Lewis, who is a student at Eckerd College. Her mentor this summer is Dr. Jason Dworkin, and she's going to talk to us about amino acids in the bag using various approaches to detect contamination in meteoritic samples and other samples.
Go to the arrow at the top. No, up one more next to Charlotte's name, and click on yours, yeah. And then go to the little over right there, the full screen, yeah.
Okay. Hi, I'm Ariel Lewis, and as Dr. Muma said, I did a study on amino acid contamination using LCFD TOF-MS, which is liquid chromatography fluorescence detector time-of-flight mass spectroscopy. And I did this study concerning amino acids due to ANSMET, and for those of you who don't know, ANSMET is the Antarctica search for meteorites. And they choose Antarctica as a site because it's very easy to see meteorites on white ice. And also, because of the way that the ice shifts, you can get specific places, sites such as Lewis Cliffs, that collect large amounts of meteorites over long periods of time. So they've been collecting these meteorites since 1976, and they've collected over 10,000 meteorites since then. And what they do is they find a meteorite, as you can see in the picture on your right, and pretty much put it in a bag, as much like the ones you see in the kit on the left. But what's happened is when you take these meteorites from ANSMET and you hydrolyze the meteorite samples, you're getting this large mystery peak in the chromatographs. And so this mystery peak was determined to be epsilon amino N-caproic acid, also known as ECA, and it's a hydrolyzation product of nylon. So this contamination source should be entirely preventable. And you can tell that it's ECA because if you look on the fluorescence detection here, you see a fluorescence peak, and the way that these are derivatized, it should be only that primary amino acids fluoresce. And so you know it's an amino acid, something with a primary amine, and then when you look at the mass spectra, you can see that it's of mass 393, and you run standards against that. So I looked at ECA contaminations, and specifically, as well as contaminations from a handful of other amino acids, because as amino acids are incredibly important to finding potentially the origin of life, and the homochirality of amino acids is something that's specifically interesting to astrobiology. And so there's 20 coded or proteinaceous amino acids, and I think we looked at about 14, a mix of biological and non-biological amino acids. And I looked for these using liquid chromatography fluorescence detection TOF-MS. And this is a schematic of the instrument that I used. And so if you start down here in the bottom right, this is the HPLC injection site. And so you inject your sample, and it goes onto the column. We used C18 columns, so this is reverse. And then the sample actually went through the fluorescence detector, and from the fluorescence to the TOF. So from this instrument, we were able to determine exactly which amino acids were in the samples. So specifically, I looked at these three bags, FR2176, A7373, and a KNF bag, which is from the corporation, Kenlam, I think. And so the way that I looked at these bags was I put clean water samples in each of the bags and left those clean water samples for 24 hours. The samples were then extracted and split, and half the sample was hydrolyzed and half of it was not. And then those samples were derivatized and analyzed with the liquid chromatograph. So these are the chromatogram results. And so in order to just point out that they're chromatograms, not spectra, it's time versus intensity. And as you can see clearly, that some bags had far, far less contamination than others, with the FR2176 showing much less contamination, particularly in the eco-amino acid. It's also noteworthy that I analyzed the bag that the KNF bag was packaged in, because I was seeing nylon peaks or ECA peaks that should not have been in the interior bag. And when I analyzed that outer bag, all these biological amino acids make sense since people were touching the bag and handling it, not like it was going to be analyzed. But there wasn't too much ECA in it. However, we think that perhaps the interior bag was heat sealed into it and it enabled contamination to be passed from the outer bag to the inner bag. So these are quantitations of the qualitative results you just saw. And if you look at the column on the left, this is the bag that ANSMET was currently using. They're using nylon bags, which puts in huge, huge amounts of ECA contamination. And I think that the FR2176 bag, which has only, I think, 4 picomoles of contamination, should be used instead. And the reason that this would be a much better bag to use is that this ECA contamination puts in a lot of extra carbons and nitrogens that would make it impossible to do isotope ratios on some of these meteorites, and making it very difficult to determine origins. So in summary, contamination by ECA is preventable. How the bag is packaged affects the bag, and FR2176 is a better bag for meteorite collection than what they're currently using. And this is the FR2176 bag on your right. So I would like to thank Jason Dorkin for being my mentor and helping me get acquainted with the instrument, Danny Glavin for pretty much coming up with the bags and giving the bags to me, Jamie, Elsa Cook, Jim Doty, and Millie Martin for showing me around the lab and helping me get used to the equipment, Kevin Ryder from Johnson Space Center for providing the bags, Corinne Eby for organizing the internship, and the NASA Astrobiology internship program. Thank you.
I don't think I'm ever going to use those bags again.
Or any bags.
Right, really.
It's also worth noting Stardust used those.
What's that?
Stardust used nylon.
Oh, yeah. Oh, boy. All right. Okay. Any questions for Ariel or comments here at the
yes.
center?
Is there
someone on the far end has a question. I heard you.
Yeah, I was wondering if there was a cost between the bags that they're currently using and the bag that you're suggesting would have less contamination per bag.
I think the new ones are more expensive, but I don't know the number. Nylon's pretty cheap.
I don't have the numbers. I could get back to you on that. But honestly, when you think about the preciousness of the samples, it's probably worth the upgrade.
Okay. I was just
Could you identify yourself, please?
I'm Sasha Rafkin. I'm an intern working here at NASA Ames.
Thank you.
Is there any plan to feed this stuff back to Kevin Ryder so that they can plan appropriately for the next expedition?
I think Danny's already emailed him.
Good.
They know.
What are these other bags made of?
The bag is made of, if I can go back. The good bag is made of polyester, low-density polyethylene foil, and polyethylene. The bag they're using right now is just nylon.
I guess none of these are Teflon?
No, Teflon actually was run as well. And Teflon had fairly high contamination levels, nowhere near as high as nylon, but still out of the realm that you want to use it.
Is there a way to... I'm just curious, is there a way to know what other groups may be using the nylon bags and tell them about your results? Is this something that you guys do?
GSC knows about it.
Yeah.
Johnson Space Center is a major.
Danny just said that JSC knows about it, and I think it becomes pretty apparent if something has been in a nylon bag once you hydrolyze it. Like, you see that peak all the time, and it's absolutely huge compared to anything else. So,
All the Apollos.
yeah, there's a conference in March that I'm going to be doing a poster on about these results as well. So.
But I think it's fair to state that the results of your research have really changed the protocol for sample collection and curation. So a very significant result. Thank you very much.
Thank you.
Okay, moving right along, we have Lauren Loudon, who's a student at Keene State College. He's been working with Richard Walker in the Geosciences Department at the University of Maryland, and working on lunar samples, particularly with respect to the siderophile elements and osmioisotopic systematics.
Thank you, Dr.
Take it away.
Hi, my name is Lauren Loudon. I'm a student at Keene State College in New Hampshire and a second-year intern here at the Goddard Center for Astrobiology. This summer I continued my work with Doctors Walker and Puchtel, characterizing the impactors of the late heavy bombardment using highly siderophile elements and osmium isotope systematics in the lunar impact melt breccia 76055. The breccia 76055 was collected from the Serenitatis Basin during the Apollo 17 mission. So we're concerned with the composition of the impacting objects that were involved with the late heavy bombardment. The late heavy bombardment took place about 3.9 billion years before present, and it is hypothesized as one of the delivering mechanisms for prebiotic organics and water to the Earth. However, there is much controversy over the composition of these objects and where in the solar system these objects formed. Lunar impact melt breccias are essentially fingerprints of the 3.9 gigaannum impactors. A lunar impact melt breccia is composed of fragments of the lunar crust, whatever else is contained in the lunar crust, and melt from the impacting object. This process retains the signatures of some elements. The highly siderophile elements, which include rhenium, osmium, iridium, ruthenium, platinum, and palladium, are the easiest to discern given their geochemical nature. The highly siderophile elements prefer the metal phase over the silicate phase, so upon core formation, they're nearly quantitatively removed from the silicate portions of the Earth. We can then take our isotopic data from some of our highly siderophile elements and the highly siderophile element ratios and plot them comparing the values to other chondrites and lunar impact melts or other samples. We analyzed 7 additional 40 to 200 milligram subsamples of the breccia 76055 by taking whole rock chunks and doing a high pressure temperature digestion on them. We then measured the highly siderophile element concentrations by isotope dilution techniques using thermal ionization mass spectrometry and inductively coupled plasma mass spectrometry. Here we have a plot of iridium in parts per billion versus the other highly siderophile elements in parts per billion. For some reason my y-axis is cut off. I'm sorry about that. Usually in the past these graphs have been interpreted to mean two-component mixing between a low highly siderophile end-member component and a high highly siderophile end-member component. The low end-member component would be considered the lunar crust, and the high highly siderophile end-member the impacting object. The slopes of the lines would then equal the ratio of highly siderophile elements to iridium in the impacting object, and the y-intercepts would equal the contribution of the target component, so what's already in the lunar crust. As you can see here, what is very interesting is we have non-zero intercepts in palladium, platinum, and ruthenium. This is unlike any other impact melt breccia we have seen in this high of a concentration, and if we compare these values to pristine lunar rocks, they usually have highly siderophile element concentrations in parts per trillion. Here's a comparison to the Apollo 14 breccia 14321 analyzed by Puchtel et al. In 2008, and we can see that within the uncertainty of our regression analysis, the intercepts would be statistically unresolvable from 0. Here we have a plot of 187 osmium over 188 osmium versus what would be palladium and iridium. The 187 osmium to 188 osmium ratio is a good proxy for the 187 rhenium to 188 osmium ratio in the impacting object. The X error bars here actually represent the range of 187 osmium to 188 osmium in the sample. The Y error bars would represent 2 sigma error from the regression analysis. In the past, we would have interpreted this breccia as diverging from the known chondritic groups, which are plotted here. We have ordinary enstatite and carbonaceous. It would be plotting diverging from the known chondritic groups, but maybe overlapping within uncertainty with some of the carbonaceous. However, what I'll show you is that might not be usable here. Here we have a plot of 187 rhenium versus over 188 osmium versus our osmium isotopic composition, 187 osmium over 188 osmium. This plot defines, or you can see there's a correlation, a loose correlation, between the two isotopic compositions, and this would traditionally define a isochron. Here I plotted for reference the 4.55 gigaannum isochron and the 3.85 gigaannum isochron. When we age correct this data back to the 3.85 billion year impact, we can see on a plot of osmium in parts per billion versus our osmium isotopic composition at 3.85 gigaannum that the isotopic composition has evolved from a isotopically uniform reservoir. This is also a very unique thing to the Spretia. So we need to know, why are there these large ranges in our rhenium, osmium, and highly siderophile element ratios? There are two possibilities, the first being two-component mixing between an impactor and the lunar crust. To have this occur, we need rhenium to fractionate from osmium during the impact and our other highly siderophile elements to fractionate from iridium. There is a possibility of volatility here, but we have reducing conditions under impact, and it requires temperatures in excess of 3000 K to fractionate or to melt rhenium. The other option is three-component mixing between a pre-serenitatis impactor, the serenitatis impactor, and the lunar crust. In this case, the only requirement is that the two impactors have different rhenium-osmium ratios and highly siderophile element ratios. Here I have a plot of what is iridium versus palladium, and this is just a schematical representation of how you may take two impactors to generate non-zero intercepts by mixing the two impactors and the lunar crust. So almost three-component mixing. And there you see the serenitatis impactor, the pre-serenitatis impactor, and the mixing line, which would give us the non-zero intercepts. These lines are exaggerated, so this is not an accurate model of the impacting ratios. And here, for reference, I plotted the palladium values from the Breccia 76055, and you can see that this is one way that we might be able to evolve non-zero intercepts in the lunar crust. In conclusion, 76055 is a very unique Breccia where platinum, palladium, and ruthenium all have non-zero intercepts on plot versus iridium. There is a range of osmium composition which reflects variable rhenium-osmium in the material. Palladium, iridium, platinum, iridium, and ruthenium, iridium ratios are much more variable than in other lunar impact melt breccias. The easiest conclusion to invoke that would result in all this is three-component mixing between the serenitatis impactor, a pre-serenitatis impactor, and the lunar crust. But it's also, we should note that there is a possibility for two-component mixing, but the situations that would need to result to have it give us the data that we see are much harder. I would just like to acknowledge Dr. Muma and Corey, everybody else at the Goddard Center for Astrobiology, and the Isotope Geochemistry Lab at the University of Maryland. Thank you guys very much.
I was actually kind of interested. Could you go back to that one slide where you showed the iridium isotopic ratios versus palladium, the palladium iridium abundance ratio? There, right there.
Yes.
That kind of looks like you've got two distinct families of points on that chart. Is that just my eyeballs getting old, or is it possible that that's real?
Are you talking these?
No, I mean the two different slopes, almost like two different slopes. If I could sort of say one set of data coming through here, for example, one mixing line, and then another one which is displaced from that up here.
Well, I think what we're having kind of problems with now is that if we have more than two-component mixing, these might not be appropriate graphs to use to show a mixing trend. And I would actually have to ask Dr. Walker if this would even be an appropriate graph to show a two-component or different mixing trends on. I know, like, traditionally or in the past we would show like a good mixing line, we would plot osmium concentration versus osmium isotopic composition, and that would give us a nice mixing trend. So
yeah, he can generate about 10 of these different plots with different parameters.
Yeah.
So some of the apparent correlations go away in the other figures.
Right. Okay. Let me go to our remote sites and ask if any questions out there. Okay. Any questions here at Goddard for Lauren? Or comments? All right. Thank you very much, Lauren.
thank you.
I should say you're now admitted to the University of Maryland for graduate study?
Not admitted yet.
Not quite.
No,
But almost.
not quite.
Almost. It's coming.
It's coming.
It's coming.
He's still working on completing his application.
Yeah, Rich Walker just advised me privately, he's not about to let him out for vacation until he completes, what, a publication?
The application.
Application. Oh, I see, the application. Our next speaker takes us from the realm of meteoritics and lunar samples to the realm of our sister planet Mars, where she has been looking at the search for signatures of carbon and life. Nadia Radova from Connecticut College has been working with Geronimo Villanueva here at Goddard on high-resolution spectroscopy of Mars. Nadia?
Hi, my name is Nadia Radova. In the summer, I worked on a high-resolution spectral survey of Mars at infrared wavelengths, and I searched for signatures of life. As an introduction, before 1970s, there were very few observations of Mars done. And here I'm showing a spectrum, an example of a spectrum that was obtained in 1963. And along the x-axis, I've plotted the wavelength in micrometers, and along the y-axis is the resolving power. Before 1970s, most of the observations were done with the resolving power lower than 10,000. Like this observation here done at 300 resolving power. And that's why the spectrum obtained showed only very bright CO2 lines, but no weak features. In the new millennium, more and more observations were done with newer instruments with the resolving power higher than 10,000. And that's why scientists were able to identify weak molecules of carbon monoxide, methane, H2O, and other molecules that are relevant for the search of life on Mars. The data analyzed this summer was taken with a NIRSpec spectrometer at KEC2, and it's a spectrometer with a resolving power of 40,000, and it has three really wide regions at the infrared, center around 1.3, 3.3, and 5 microns. Due to the fact that the range is really large and the resolving power is high, the area under every region is very large, and this means that we can detect many weak lines and retrieve very precise abundances of the molecules observed. This is an example of the spectrum we observe, and thanks to the fact that the resolving power is 40,000, we can observe many weak lines of carbon dioxide and even weaker lines in between. In contrast, the same spectrum taken in 1963 with the low resolving power showed no such lines, and only very bright lines. The data analyzed this summer was taken with the NIRSpec spectrometer at KEC2 in Hawaii, and all the data is taken along a slit positioned along Mars, spanning from north to south. And all data comes in images like this, where every order is a spectrum at different wavelength, and it spans from north to south along the slit. All the spectra contain terrestrial, Martian, and solar lines, and my task this summer was to remove the terrestrial and solar lines in order to obtain only the Martian lines. The data that I analyzed was taken on March 21st and March 22nd, 2003, with 4 of the NIRSpec settings. With the NIRSpec 3 setting, I identified water, carbon dioxide, and O2 single delta molecules, as well as solar lines. The M-wide setting center around 5 microns showed carbon dioxide, water, and carbon monoxide molecules. The KL1 and KL2 settings are important because they include a wavelength of 3.3, which is the order here. And in these settings, we can observe many hydrocarbons, which are relevant for the search of life on Mars and any biomarkers on Mars. In order to process the spectra, I used 2 programs developed here at the Goddard Astrobiology Center. For data processing, I organized and cropped all the orders of interest, cleaned bad pixels, and straightened the orders spatially and spectrally. And after that, I calibrated the frames and stored the results. For the residual analysis, the program let me define the observing geometry to calculate local atmospheric conditions and extract Martian residuals as well as the molecular column densities. After all my work this summer, I plotted all spectra in graphs like this, where along the x-axis I plotted the frequency in Mars rest frame, and the y-axis is the normalized absorption spectrum. In black here I show the observed spectrum that contains Martian, terrestrial, and solar lines. Red is the model of the terrestrial lines we use for the program, and when the two are subtracted, we receive the residual, or these are Martian lines as well as solar lines. Here in this order, I identified carbon dioxide branch, as well as water molecules and solar lines, and for all orders, I identified the type of each solar line observed. I also confirmed a new CO2 band that my mentor Geronimo Villanueva found, and the new lines are plotted in red. It's important for us to identify unknown features because they'll let scientists not mistake carbon dioxide with methane or other weak lines that are biomarkers. These are two orders that are used to retrieve water and O2 single delta abundances on Mars. The order here shows very bright water lines, and this is the order that has the greatest abundance of O2 single delta on Mars. And this is the plot that I created. On the x-axis is the latitude of Mars in degrees, and the y-axis is the molecular line intensity. In blue, I plotted the water vapor, which is very high in the northern hemisphere because the data was taken at the time when it was late summer on Mars. And O2 single delta is plotted in red. O2 single delta is important because it is a sign of the existence of ozone, because after photons hit the ozone, it falls apart into O2 single delta, and a photon at 1.27 microns is emitted. And the plot here shows the expected anti-correlation between water vapor on Mars and O2 single delta on Mars. And I would like to thank the Goddard Center for Astrobiology, Dr. Muma, my mentor Dr. Villanueva, Corinne Eby, and all summer interns.
Thanks very much, Nadia. I wanted to ask you briefly, it was a great deal of information, and I wanted to ask you, in your opinion, what is the most valuable thing you learned this summer?
I think how to process all the spectra and how to retrieve all Martian lines, and how to remove all the terrestrial and solar lines that we observe from the Earth atmosphere, because Keck-2 is an Earth-based telescope. You have a lot of contamination from the Earth atmosphere.
Well, you can feel very satisfied with what you've done, because this is a great challenge to do this so well. Yeah.
Thank you.
Anyone else here wish to ask questions?
How do you extract the Mars rest frame correction? You have an x-axis saying Mars rest frame?
Yes, we use the Doppler shift, and we correct all the lines with the Doppler shift.
So which direction is it? Red shifted or blue shifted?
Not sure.
Okay.
It's minus 15 kilometers for this.
Did you see
And that's
any...
based on comparison with the rest frequencies for known lines, such as water vapor and so on.
Yeah.
Did you look at all at the hydrocarbon variance with latitude, like you did with water, or no?
No, I did this only for water and O2 single delta.
Are there any questions on the remote sites? Okay. Thank you very much.
Okay.
Our next speaker is Lily Raines, a student at Eckerd College. She's working here this summer with Marla Moore on issues related to the carbon structure, chemistry, and spectroscopy of frozen ethane.
Good afternoon, my name is Lily Raines, and as Dr. Muma said, I studied the structure, chemistry, and spectroscopy of frozen ethane. One of the reasons that ethane is so interesting is because it is found in a wide variety of solar bodies in the solar system. It's been found on Titan and on many trans-Neptunian objects. For example, it's been found on Oort-Cloud comets, Comet Hyakutake, which is shown here on the right, on Pluto, Quaoar, and 2005 FY9. A spectrum from 2005 FY9 taken in the near IR that shows the presence of ethane is shown here. These three peaks are distinctive of ethane. In addition, ethane formed from the irradiation of methane, which is abundant throughout the solar system. Therefore, it's reasonable to believe that we could potentially detect ethane in any location where you could find methane that is exposed to radiation. In addition, most A values that are currently used to relate peak intensity to the number of molecules in a given sample for ethane are based on crystalline ethane only, and there have been very few investigations into ethane's behavior in the near IR. This data would be particularly useful because the near IR generally does not suffer as much interference from Earth's atmosphere, and therefore Earth-based observatories could use this data to detect ethane in other bodies. We studied the properties of ethane under changing temperatures and radiation doses this summer. We did this by forming ice samples by condensing gases onto a small substrate that was cooled to the desired temperature, as seen here. We typically deposited our ethane ices around 10 to 60 Kelvin, and we took IR spectra periodically during our ice formation and while changing the temperature and radiation dose. Our radiation was in the form of accelerated protons generated by a Van de Graaff accelerator. Shown below is a typical spectrum of amorphous ethane. Highlighted are the regions in the near IR from about 4,500 to 4,000 reciprocal centimeters. These bands are very weak, but they're very distinctive. And shown here are the peaks around 1460 reciprocal centimeters in the mid-IR. The peaks in the mid-IR were much more cleanly separated and much stronger than those in the near IR. Because these peaks were clear, we were able to determine A values for metastable and amorphous ethane ices. A values in this case refers to a way to relate peak intensity to the thickness of a sample, or the number of molecules present. To do this, we measured how band area increased with the increasing thickness of an ice, and this data is shown here with band area across relative thickness for 4 different bands in metastable ethane, and these values are shown here. These same calculations were done for amorphous ethane as well. The peaks in the mid-IR around 1460 reciprocal centimeters are also interesting because they are the most distinct between the 3 different phases of ethane we studied this summer. As you can see in here, we have an amorphous ice, a metastable ice, and a crystalline ice, all being stable at 14 Kelvin. Shown here are spectra of ethane in the near IR, again for these 3 different phases. Unlike in the mid-IR, these peaks are very similar across the same 3 phases, except for this one around 2.5 microns or approximately 4,000 reciprocal centimeters. This is the only really accurate way to tell in the near IR the phase of an ethane ice. Shown here is one of our amorphous ice spectra overlaid with the spectrum from 2005 FY9 shown earlier. As you can see, these 3 peaks do correlate with these seen in FY9. However, the peak that identifies the phase is not shown. So, future Earth-based observations, if they include this peak, we could potentially determine the phase of ethane present on a body. These 3 different phases of ethane are formed by depositing ethane ices at different temperatures. Amorphous ices form between 10 to 25 Kelvin, metastable ices form between 25 to 50 Kelvin, and crystalline ices form between 50 to 60 Kelvin. Metastable ices are particularly interesting because this is the temperature range that most Trans-Neptunian objects similar to Pluto exist at. These are typically they're about or less than 40 Kelvin. So we would expect to see metastable ices on these bodies. Shown here, this demonstrates the thermal properties of these ices. They will retain their structure when cooled. However, when they are heated, they will become crystalline irreversibly. Shown here on spectra A through E is the same sample of ice, deposited at 50 K, cooled down to 14, rewarmed, remains metastable. However, as soon as it's heated to 65 K and then cooled back down, it will remain crystalline. In contrast, when these ices are exposed to radiation, they have a tendency to become amorphous and to produce many different hydrocarbons as radiation products. Shown here is a sample that was exposed to about 10 electron volts per molecule of accelerated protons. In conclusion, this research we did over the summer determines that the differences between amorphous, metastable, and crystalline ices in the IR spectra, both in the near and mid-IR, are significant and potentially useful for Earth-based observatories. Ethane ices will retain their structure when they are cooled. However, they will become crystalline when heated to a sufficient temperature. But these same ices will become amorphous when irradiated. Our improved understanding of ethane may contribute to future searches for hydrocarbons in the outer solar system, particularly missions similar to the New Horizons mission to fly by objects and obtain spectral data. I would like to thank Dr. Marla Moore, Dr. Reggie Hudson, and Dr. Zahn Peters for all their help this summer, Steve Brown, Tom Ward, and Eugene Jeroshenko for the operation of the Van de Graaff Proton Accelerator, Karina Ebby and NASA for this great opportunity, and I'd like to thank you all for your time.
All right, I think we'll take questions from the remote sites first. Anybody out there want to? Yes.
I was wondering what the measure of significance was for spectra. Is it qualitative, where they look different as we saw, or is there a quantitative way to say that two spectra are significantly different?
Well, mostly we relied on quantitative differences because, as you can see around the peak, around 1460 reciprocal centimeters, it's very distinct. You can very clearly quantitatively tell the phase of the ethane. However, we also did measure band areas, and we figured out the relative intensities between these peaks, so we can have a good idea what phase the ethane is in.
Thank you.
Yeah.
Around the room here, anyone? I wanted to ask you about your comparison of your laboratory spectrum of the three ice phases, the infrared spectrum, with the spectrum of the remote, I guess it was detached object in the Kuiper disk.
Oh, yes.
Yeah, and you indicated that the amorphous, the signature that would identify amorphous ice was not seen, and it wasn't clear to me whether you meant it hadn't been tested or was tested and wasn't there.
Well, we're not sure. From reading this paper, it's hard to tell if they did take the observation out to that wavelength or not.
Okay, so, that's clearly suggests some future work is needed.
Definitely, yes.
Very good. Okay, thanks very much.
I do have one question, too.
Yes.
You have the amorphous, the metastable, and the crystalline ice, and you showed that with the metastable, when you went to higher temperatures, you got the crystalline and came back. Does the amorphous go to the metastable, or does it go straight to the crystalline?
No, actually the amorphous goes to the crystalline, and it goes at a temperature much lower than the metastable ice requires.
Okay.
So you can only form the metastable ice if the ice is formed
it there.
at that temperature. Right.
Okay.
All right, thank you very much. Next, we're going to go outside the solar system virtually and listen to Kamen Todorov, who has been working with Greg Deming. Kamen is a student at Connecticut College and will talk to us about the atmosphere of a recently discovered exoplanet as measured with Spitzer.
Hello, my name is Kamen Todorov. I'm a mentor of Dr. Dave Deming. This summer I worked on observations from the IRAC instrument on board of the Spitzer Space Telescope, and we looked at secondary transit of exoplanet HAT P1b, which is a transiting exoplanet, and we did that to study its atmosphere. First, why do we want to study exoplanets? Studying exoplanet tells us a lot about formation and evolution of planetary systems in general, not only based on our solar system. We also developed techniques that would allow us to study much smaller exoplanets when observations, etc., are available with more sensitive telescopes. So far, what we know about HAT1 is that it's transiting exoplanet, which means that it eclipses its own star, and it orbits a G0 main sequence dwarf. The distance to Earth is 140 parsec. There's 4.5-day orbital period. The semi-major axis is around 5% of that of Earth. It's about half the size of Jupiter. Well, half the mass of Jupiter and a bit bigger than Jupiter in terms of radius. And all these were determined by Bakos from Harvard, who discovered the planet, and then they were improved later. But we don't know anything about the atmospheric properties of this planet, so we want to know more about them. So we looked at the secondary transit, which is the time when the planet is behind the star. At this time we can notice a small drop of brightness in the infrared where because we lose the black body flux from the planet itself. We can detect these, for instance, with the Spitzer Space Telescope, which is an infrared telescope. And what we do is we perform series of observations over about 6 hours. We measure the brightness in it, and we plot it versus time, which gives us a light curve. This is a sample of our data. Planet B, the smaller, fainter one of the two, is the HAT1 planet. And this is a zoomed view, and this is our planet, our star. And the planet is obviously unresolvable. So the first thing we did was to clean up the cosmic rays, which are faint, bright, pixels in the white circles. Then we integrated the pixels around the aperture box of the star, and that gave us a measure of the brightness of the star at that moment. And then we used fairly conventional methods of determining the background levels, removing the other instrumental effects. Our error analysis was based on a bootstrap Monte Carlo simulations, which is a well-established method in cases when the exact distribution of the points is not known. So basically what we do is we take our data set, we fit our model, best fit model in it, then we take the differences represented by the red straight lines, and then we just mix them around in time, and then we add them back to the best fit model. This gives us a new simulated data set, which we can later fit another model to it and then measure the eclipse again. We repeat that about 1,000 or 2,000 times and take the standard deviation of the simulated eclipse depths to be our standard deviation. And this is my result. So the IRAC instrument has 4 wavelengths, and I analyzed all 4 wavelengths. The 8 micron, 5.8, 4.5, and 3.6. The data is binned, so each point represents 20 actual data points. And this is a plot of time, essentially, versus relative brightness. And as you can see, we get different eclipse depths at different wavelengths. This is significant because, as you can see from this plot, which is basically wavelength versus eclipse depth, that this is essentially a broadband, very low resolution spectrum of the atmosphere of the planet, because the eclipse depth is essentially a measure of how bright is the exoplanet. These points are actually another planet, HG 209458, which is very similar to HAT1, except it's actually a bit closer towards its star. This was analyzed earlier this year by Heather Knudsen. So what they did was they fitted a model of the spectrum. These are spectrum models. The red spectrum is a model in which the atmosphere doesn't have a temperature inversion, meaning that the lower layers of the atmosphere are hotter than the upper layers. And as you can see, this model doesn't fit very well. However, the blue model, which includes a temperature inversion, meaning that the upper atmosphere is much hotter than the lower, fits a bit better, and HAT1 has similar structure to its similar structure of its spectrum. And this plot just shows what would happen if we brought HAT1 a bit closer to its star, and you can see that it's actually very similar and well within error bars to HG 209458. So this implies that HAT1 also has a temperature inversion in the atmosphere. Thank you. I want to thank Dr. Deming, Dr. Harrington from UCF for taking the data, to Dr. Muma, to Corey Abbey, and to the NASA interns. Anyone has any questions?
Floor's open for questions.
Yes.
I'm curious about the bootstrap simulation. Was code for that something that you came up with, or which program or code or package did you use?
I wrote the code. All the analysis code is written by me, almost all.
We only accept the very best students here in our internship.
Which language did you use?
IDL.
Okay.
Okay, how about around the room? Anybody here want to pitch in? Wait a minute, we can't let it get off this.
Can you just say a word about what's IDL? What does it stand for?
IDL is Interactive Data Language, I think. It's essentially a programming language that allows you to do real-time analysis as well as write scripts for it. Which basically means that you can type commands and analyze it by just typing commands, or you could write a script and just run the script, and which would do things for you.
So you've got a planet that is substantially larger than Jupiter, but it's about half the mass, is that right?
Yes, it's very close to its star, so it's much hotter. And this supposedly that makes it bigger because it's essentially the
thermal gradient.
Yeah, thermal expansion, essentially.
The largest deviation was at long wavelengths in both cases. Why?
And the noise is biggest.
Well, but they were consistently low. I mean, your models basically went nicely up. They were relatively flat. None of them had a bend in them.
Oh.
The 3 points are, you know.
Well, this is the best model that the people from Harvard came up with.
So there's a lot of room for improvement.
Yes.
Okay.
Nobody knows the answer to the question.
Okay.
But methane which has a band at that 8-micron band, was not the answer.
No.
Did they include methane in model atmospheres? They obviously screwed up. Or something. Something's different. Always believe the data, right?
I mean,
yeah.
There does seem to be a difference there.
Yeah.
No one really understands it. The 5.8 micron, there's a strong water band there. So maybe that the temperature
could be a little higher, yeah.
Even more elevated because of that strong band.
I think Inga had a question or comment. No? Okay. Anyone else? Okay, that concludes our formal talks, but I first want to, before we sign off here, and thank everyone for participating and listening, I would like to give special recognition to the 6 mentors who worked with these young people today during the summer, and as you can see, the result has been extremely good for everyone concerned, I think. So I'd like to give a hand to the mentors themselves. Last, perhaps most significant, I wish to draw special credit to give special credit to Corey Eby, who's about to zoom in on herself.
No, I'm not.
who has really done an outstanding job in shepherding everyone, and indeed throughout the summer, and making everything work. So Corey, it's a big hand. Now, unfortunately, the folks in remote sites don't get to share in the goodies that we've laid out on the other side of the room here, but we hope you'll have a snack wherever you are.
We're about to go have lunch. I just wanted to say real quickly that, as always, all the students were excellent, very good, high-quality presentations, very articulately given, and I want to say thank you for all your time, and Corey and Marco, the talent behind the curtain, thanks for all their good work on this as well.
And these will be posted on the NAI website in a couple days, so you can view these over again with the slides and the video.
Good. We had one question about that before we started today. One of the students, I won't mention who it was, but she wanted to send it to her mom or something. Maybe look at it 40 times to see how it could be improved. Okay, thanks a lot, folks. We'll see you all soon.
Thank you.
Have a good day.
Bye-bye.